So I wanted to kind of set the stage. So why does it matter what, for example, myself and others, in the field are doing as a potential therapy for the future? Well, the reason why it matters is very important. It's because we are projected to be, in about 30 years from now, 10 billion people on this planet. 10 billion people, maybe slightly more, slightly less. 20% of them, me included, will be 60 years old or older, okay? So 2 billion people on the planet, okay? So what does it mean? Well, it means that, overall we're gonna be a very wise kind of, population, right? What it also means is that 2 billion people will be at risk of developing one or more conditions which are associated with aging. And aging, as we have heard, is the #1 risk factors for those conditions. So we develop those conditions because we get older, all right? What can be done today, what could be done tomorrow, but fundamentally, even if we do, we're still gonna age. We're still gonna deteriorate with time. So the question is, can we do something beyond that? All right, so that is what we are trying to do, okay? So let me take a step back, because again, I want to explain you very carefully what I'm gonna talk about. So regardless of whether we are young or we are old, okay, the reason why we live is because there are very special cells in our body. They're called stem cells. They're very rare. They do not exist in every tissue. Some tissues have a lot, skin, intestine. Some tissues have very few, for example, the brain. Some tissues have none, the thymus and the heart, for example. There is no stem cells, at least up to now. We haven't been able to discover them, right? So it is because of stem cells that we live as long as we do. If we didn't have stem cells, you can see it right here. We would lose our intestine in 2 days. It would be gone. We would lose our skin in 3 weeks. Would be gone. And we would lose our blood cells in about 4 months. So stem cells are there to regenerate, right? Are there to replenish what gets damaged, what gets lost, and we lose millions of cells every single day, all right? So stem cells are there. They're very rare. They're very quiet, very shy. They typically don't do much, right? They just reside in the tissues, and they're just waiting for signal from the outside, from the body. Okay, now it's time for you to do something, because, for example, we got a damage, we got a wound, something happens, an accident, a broken bone, whatever that is. There is a signal for them to say, okay, it's time to regenerate the tissue that you are residing in, all right? So that's what stem cells do, okay? They are, again, rare, somewhere in the body, and their function is just to regenerate the tissue. So unfortunately, to go back to one of the questions, like, why don't we live forever? I mean, we have a perfect system here. Why we don't live forever? Well, unfortunately, we don't live forever because of what Marco and Maddalena have said already, but also because, in addition to what they have said, there is a phenomenon which is called epigenetic drift. It means that progressively, when they divide the cells, they are not identical to what they were before. Every time they divide, they accumulate damage. Every time we drink too much, we don't sleep enough, we get stressed out, we yell at our partner. Every time something like this happens, there is damage coming from the outside that basically signals to the nucleus, to the central core of the cells, and there is a progressive accumulation of damage. That's why we age, in very simple terms, of course. So this leads to deterioration of stem cells, organs, tissues, and so on and so forth. So what are the possible solutions for the day after tomorrow, okay? So one solution is that we can generate new stem cells, new tissues, new organs, or forget about that, we can rejuvenate your existing stem cells, your existing organs, your existing tissues. Okay? So that's what I'm going to talk about. So how do we do this? Okay, so let me take a quick step back. So we have learned that there are stem cells, there are other cell types in our body, okay? Now, a few years back, about 12, 15 years ago, a Japanese scientist, he's called Yamanaka, probably you have heard about him, made a brilliant discovery. He figured out that actually you can take, let's say you can take a skin cell from your arm, okay? You can take that cell. So what that cell does is just, it's a skin cell. It doesn't do anything else, all right? You can take that skin cell and you can reprogram it, okay? So you can change its identity to an embryonic-like state. So you can make an adult cell and you can turn it into an embryonic-like cell. Why is this so brilliant? Because embryonic cells, because they are embryonic-like, they can differentiate then not only in the original skin cell type, but also into any other cell type of the body. Brain cell, liver cells, blood cells, bone cells, you name it. All right? So this is brilliant because now, virtually from any person, from each of you guys, we can make embryonic-like cells from your skin cells, and now we have a source, potentially, of organs, tissues, that can be used for, for transplantation, for example. So so this process is very long. It takes about 3 to 4 weeks on average to go from here to here. And then, depending on the cell type you're dealing with the organ you're dealing with, it may take another 4 weeks or months. And so there's still a lot of work going on here, and this is one of the challenging factors. So prolonged reprogramming gets you here, all right? So the good thing is that they are unlimited in numbers, so once you make them, they are immortal, but immortal in a good way, not in a cancer way, okay? So they're immortal in a good way, so that means that you can make as many as you want, okay? And so you can really generate a lot of cells from them, right? They are genetically matched, so that means that, if I make iPS from you, they're called iPS. If I make iPS from you and then I make a liver from those cells, the liver is genetically identical to you, so you're not going to have immune rejection. So they can use, as we said, to regenerate any cell of the body, and in the near future, and I'm the optimistic guys if you didn't figure it out yet, but in the near future, from those cells, we will be able to bioprint tissues, organs, and some people are actually working in generating, using the same cells, organs in large animals, for example, pigs, sheep, and so on, for transplantation, okay? So right now it's kind of still early stage, so it's kind of, discovery, R&D. No. They're doing it in pigs and in really large animals. Yeah. It needs a very, very sophisticated approval from the ethical committee, of course, because they're not using primates because it's way too expensive. So they're using pigs, for example, sheep, goats. So here in the United States, yes. Human organs, yes, in animals. I know the ethical question is coming. Okay, we can keep it for later. Okay, so what we have discovered in my lab, and this is the foundation then of Turn, is that when you do this reprogramming, you go from an aged skin cell to a youthful embryonic cell. So you have two things happening: change in identity of the cells and change in cellular age, right? What we have discovered is that if you stop the reprogramming, so if you stop the expression of this, let's call it magic cocktail, early enough, the cells don't change their identity, so a skin cell remains a skin cell, but it's just a more youthful version of it, okay? So short reprogramming does not impact cell identity, but it does impact cellular age. And so, a long story short, I'm envisioning a future, and we have already shown it, again, at the proof of concept level for now. There's still a ton of studies to do. But we have shown that actually by pulsing the expression for a short time of these reprogramming factors that work on the epigenetic drift problem that we talked about before, you end up with a youthful cell. And it's youthful by all means. Not only youthful because it's epigenetically youthful, but it's mitochondrially youthful. It can interact with the environment in a more youthful fashion, and so on and so forth. But it's still the same cell. So now the idea would be that if this works in vivo, for example, there is no way for me to take your skin cell, reprogram them to iPS, make a liver, and then transplant it. I can go directly into your liver and make it younger. So that's the idea. Okay, we are far away from that, but that's the idea. Okay, and so basically, this is what, you know, kind of a nice rendering of what I talked about. So we're really seeing, and it's not working, of course, but we're really seeing a future where basically we would be able to reprogram, rejuvenate, make the cells younger again. I don't think that there's going to be one solution. There's going to be many different solutions, and we should actually use them all for the common purpose, which is enhancing health span. That's what I think. So I have to make a correction. Overall, what you have said is correct, but there is just a small imprecision there. Actually, the germ cells, so the sperm and the egg, actually those are the cells that do not age. So that's one of the reasons why I believe that at the biological level, immortality is possible, because the germ cells are the link between myself, my kids, my grandsons, and so on and so forth, and are a connection between myself, my parents, my grandparents, because there is a continuum that doesn't end. So there is an immortality of life, okay? So you said humankind is aging. No, as a species, you know, Homo sapiens, like any other species, is not aging. The species doesn't age. It's the individuals within the species that age, okay? So I don't know if, I personally don't think there is a design, but it doesn't matter. Life is immortal. We are connected through our germ cells, again, as I said before, we are connected with the earliest forms of life that showed up billions of years ago on the earth. We are connected to them. So life is immortal, okay? Yes, but there are mechanisms, and the work that we have been doing is actually inspired from those mechanisms. There are mechanisms that prevent aging from being transpassed from one generation to the following. There are embedded mechanisms in reproduction that allow whatever mistake is made, and again, I'm talking, you know, broadly, okay? There are always exceptions, okay? But there are mechanisms in the germ cells and in the embryos that kind of wipe out the epigenetic mistakes that have been accumulated in the meanwhile. And that's why there is a perpetuation of life. So we do it by, so how do you reprogram the cells? So basically you provide the cells with genes, okay? So these are, the genes are basically instructions, you know, in the DNA that tell the cells what to do, okay, in very simple terms. So basically we are providing the cells with those genes in a form of RNA, it's called RNA. So we are so we are really injecting inside the cells those mRNAs that typically are not there. And those genes are so powerful, you know, that they go into the nucleus and they start reorganizing, rewiring, restructuring the chromatin in ways that, again, we are still far from understanding, but in ways that seem to lead to a more functional, youthful state.
So do you use mRNA technology?
Yes,
okay.
Because mRNAs are relatively simple to make, okay? they don't change the genome of the cells. So when you inject an mRNA into a cell, the cell remains genetically unaltered. If you inject DNA into a cell, the DNA can integrate into the genome and can change it, sometimes in unpredictable way. mRNAs can't. mRNAs just go in, and what they do is make the protein that they are supposed to make, and the protein does the job. As simple as that. So why is this important? Well, again, they're easy to make. Technology. We are learning how to print mRNAs at very, very large scale. So this is going to impact the cost of the mRNAs. It's going to drop it down dramatically. It's going to make, medicines, mRNA-based medicines, very affordable, very democratic, okay, for everybody. It's going to scale up the production kind of capacity, okay? Because at the end of the day, it's going to be, like printers of RNAs that are going to just work, and just generate mRNAs and, in a limited fashion. So this is going to really impact, and I think that the future of gene therapy, and gene therapy goes beyond what I've told you. Gene therapy is the ability to provide a gene that is missing, whatever that is, the spike protein for COVID-19, or whatever that is. mRNAs are going to actually be the revolution, for the future. So yeah, it's a good example of when technology meets science, and there is an inflection point all of a sudden.
Thank you.